Well, good evening, ladies and gentlemen, colleagues and friends. You may be wondering why I'm not here tonight. I'm actually with Christina Curtis at a very important AACR symposium in Washington, D.C., where we talk about colon cancer stem cells. And this is very close to my heart because only when we learn how to deal with colon cancer stem cells, we have a chance to cure colon cancer. So you can understand I have to be there in order to find new ways to treat colon cancer, in order to really unroot colon cancer by targeting the colon cancer stem cells. The good news is I have incredible people who represent me, and it's a great honor that Dr. Afsane Basi will talk about her kind of research in colon cancer development, prevention, as well as screening. And one of my incredible postdocs from Italy, from the University of Padua, the Center for GI Cancer in Europe, will talk about the clock proteins, which I'm sure you find fascinating. It's my big pleasure to give you an update what we have achieved over the last year with the help from the WunderGlo Foundation and focusing on the goals of the Wunder project. We have made incredible progress to better understand colon cancer. And when I mean better understand, we are really unraveling the genetic secrets and mystery of colon cancer in order to better develop more successful treatment opportunities. The largest clinical trial ever done in the world for metastatic colon cancer was conducted in the US, where I am the PI and oversee all translational research. We have collected over 40,000 samples starting over 10 years ago. And this is now being worked up on a genetic level which is unheard of. We have done gene expression levels, we have done DNA mutation, we have done genome-wide association studies, we have done RNA-seq, and we are just waiting any day right now to hear from the Moonshot program from the NIH to see if we can do whole genome sequencing on the tumor and methylation sequencing on the tumor. Knowing and having these additional data would give us an incredible new insight why certain patients have more aggressive disease or respond to specific treatments than others. So this will change the way we look at colon cancer and will be a change in the paradigm. Now, in order to make this work, we worked very closely with our collaborators in Europe, particularly Sabine Teschper, who is actually on our advisory board, from Belgium, one of the leading actors in Europe to do molecular characterization of colon cancer. And the PI of the second largest study ever done, Dr. Sebastian Stinzig from Munich in Germany, who actually spent 2 years of his career in my laboratory. And we have together now incredible, powerful tools and colon cancer samples to really find new solutions in our treatment strategies for this disease. In our effort for this large clinical trial, we have now the largest collection on liquid biopsies. USC and our team has played a significant role in developing technologies to look for tumor DNA and RNA in the blood of patients with colon cancer, mostly with metastatic disease, but we are now interested in patients who had successful surgery as the most sensitive technology to detect disease left behind, not visible on any imaging, including CT and PET scans. We are actually getting the data by the end of this week and will present them at a large meeting in January in San Francisco, one of the biggest GI oncology meetings in the world. You probably remember Christina Curtis, who studied at USC, who is also on our advisory board, and we lost her to Stanford but we continue to work together. And her area of expertise is very unique because we know colon cancer cells among each other differ significantly. Like when you put 10 people together, every person is different, so are colon cancer cells. Our goal is to find the bad ones among all cancer cells and better understand why these single cells are so bad, to have a better handle on how we can treat them better. This single cell molecular classification will be a big breakthrough in understanding colon cancer. Our data, which we did in collaboration with our collaborators in Italy, Germany, and Spain and Austria, is now being submitted to Nature, the biggest scientific journal, and it's in review, and hopefully we can publish. And for the first time ever, we can now, with single-cell molecular classification, predict if a tumor will be metastatic or not. It's unbelievable new information and will change the way should we resect the tumor or not, and how potentially treat earlier than before you see the metastatic sites on imaging and monitoring visits. So this, you can understand, we are actually making significant progress in better understanding the disease. Now, there was also a significant announcement at the ASCO meeting this year. And one of the announcements for colon cancer is that the screening guidelines have changed. Now the recommendations are to start at age 45. USC and its Department of Cancer Epidemiology were critical in understanding that overall the incidence of colon cancer is going down in the United States, but the incidence in the age group under 40 is significantly increasing. And we still do not know why. In order to capture more patients at early age, the screening years is now down to 45 years. So on a big global scope, we are working effectively together with all the groups necessary to allow really productive collaborations. I wanted to give you two examples what we do here at USC in my laboratory. We have incredible new insights and potentially new treatments in development. We realized that particular people who work night shifts develop more colon cancer. So the regulation of day versus night is extremely critical for colon cancer risk. Why? What regulates day and night? The identification of the so-called clock proteins is a new area of research which will or has the potential to change and develop new treatments. Now, one of the pioneers in the identification of clock proteins is Steve Kay, who was recruited from Scripps to USC 2 years ago. Now we work very closely with his laboratory to develop treatments. And he actually has developed a drug which works in preclinical models for prostate and leukemia. We will do it for colon. When he saw our data, how clock proteins predict outcome, he was incredibly impressed. And we will push forward to use his compound in our PDX models, tumors which we collect in the operation room from patients and put it in our mouse. This is the closest model we can test new drugs which would mimic basically the effect in human patients. A couple of years ago, I told you about we identified a completely new marker. It's called DSCR1. And we found it more by an accident because it predicted the efficacy of an antibody targeting tumor blood vessel formation. It stands for Down syndrome gene. Now you would probably ask yourself, what does Down syndrome has to do with colon cancer? Well, you should know patients with Down syndrome have significant less cancer compared to all other patients. And the reason is this particular gene, because it's a heavy regulator of tumor blood vessel formation. So we identify this gene, we now crystallize this protein, and we have a protein which is responsible for the binding to have the effect on the tumor blood vessel formation. We are working with basic scientists from the USC main campus and making very good progress. It takes all its time, but as soon as we have this protein, we will test it in our really sophisticated animal models. All these projects would be not possible without your funding and your help. We can take high-risk, high-impact ideas and push it quickly through. This is how we change colon cancer research. Since the funding through NIH is very slow and they want to see that it really works, we cannot just say we would like to do that. We are in significant need for funding for these kind of high-risk, high-impact projects, because I think that is the way to make significant change and progress overall. Please don't forget the call to action. I think the project of the clock proteins and the Down syndrome gene protein are incredible ways to support this breakthrough research, because only with your help we can make sure that we get results and get data leading to new drug development. We are all very excited about it. I'm sure you can feel it and hear it and see it, but we need your help. Without you, we cannot do it. Push the number 9. So I would like to thank you for all your support, and I hope you have a wonderful evening. Enjoy the gala and the silent auction, and hopefully we will see each other next year. Thank you again and have a wonderful evening. Good night.